Sounds good. All right, thank you very much. So like Chris said, I'm going to give 2 talks, and my first one's going to kind of wrap up what we know about Phytophthora rubi, and that's been from research that we've conducted over the last couple years. And then I'll quickly transition into some hopefully newer ideas on maybe how we can deal with pre-plant management of soil-borne pathogens and nematodes. So for those of you who haven't heard me talk before, I'm a nematologist, not a plant pathologist, but I'm representing the group that worked on Phytophthora rubi since none of them could make it. And therefore, I want to point out my collaborators on this project. And most importantly is Jane Stewart. She was a postdoc down in Corvallis, and she did the majority of this work, and none of this would have been possible without Jane. Duncan and Javier did a lot of the field and laboratory work associated with this. Tom Walters and Julie Pond helped us find collection sites for P. rubi. Nick Grunwald, he's the Phytophthora expert on this project. And then Peerbout Crop Management, who's been involved with this. So just quickly, a presentation outline. Why should you care? I'll tell you a little bit about Phytophthora rubi. Probably most of you already know this, but just to refresh your minds. I'll present our research results and then kind of some ideas of where we're going to go with the knowledge that we have. All right, why should we care? I think you probably can all attest to the fact that raspberry root rot is a significant concern to the raspberry industry. We also know that it's going to become a lot more difficult to use pre-plant soil fumigation to manage this soil-borne pathogen, and Tom will give you a little bit more background on what I'm talking about here with fumigant management plans and buffer zones and that kind of thing. Additional regulations with chloropicrin likely, so the common soil fumigant used for pre-plant fumigation is Telone C35. In that combination, 1,3-D is the nematicide, chloropicrin is the fungicide. I just spoke with Tom, though, at lunch, and I put this up here because chloropicrin has a lot more restrictions in California than it does here, but he felt like there probably weren't going to be a lot of additional concerns with this product in the state of Washington. We also know with soil-borne fungi and oomycetes that repeated use of post-plant fungicides can lead to resistance. We certainly saw this with the botrytis earlier today. And then finally, very few resistant varieties are available. Obviously, Cascade Bounty from Pat Moore's program is a variety you can use, but for the most part, there isn't a lot of resistance available. So just a little bit about the biology before I show you the results. P. rubi causes root rot of raspberry. Several multiplication cycles can occur every year, so it's what we would call a polycyclic disease. It just kind of keeps on going and going as long as the conditions are favorable. It's most active when soil temperatures are pretty cool, 50 to 60 degrees Fahrenheit. It's common in areas of field that are not well drained, although Jerry Weiland told you earlier that we do find it in areas that are drier and you kind of wouldn't expect to find it. And then plant symptoms, which you probably are very aware of. Wilting and dying of floricanes, usually in June. The lesions at the base of the cane, which I have a picture of here. You can see the lesion on the right-hand side, similar to what Jerry showed you earlier during his survey, collapsing plants. And then down on the bottom you have a collapsing area of raspberry with a little, what we call an oospore, which I'll show you in the life cycle of the pathogen. So just quickly, and this is mostly so that you kind of understand some of the challenges of managing this disease. I'm going to start down on the right-hand corner looking at the oospore. This is a resting structure. In addition to the chlamydospore, which is on the other side of the slide, the oospore can germinate, again, at low temperatures, 50 to 60 degrees, creates a sporangium. This is where these little zoospores are produced. They're released, and they swim through soil. And again, it's usually in waterlogged conditions at lower soil temperatures. They'll encounter a root, become encysted, invade the root, and then there'll be this mycelial growth in the root, and then you can see where it starts to move up and cause lesions in the cane. And I have down on the bottom, several hundred oospores can be produced per inch length of infected roots. So you can imagine if this happens multiple times, you're going to get a really rapid buildup of this pathogen in a field. Okay, so the objectives of what we did, and I just want to point out, this was all funded by a federal grant from USDA NEFA, so that's what we had, a 5-year grant with Tom Walters, and all this research was done under that grant. Our objectives, which I'll go through in my talk, first we wanted to determine the best method to detect P. rubi in raspberry, and I'll tell you why. We wanted to identify the Phytophthora species associated with raspberry, so there was a lot of not a lot of confidence that we were only dealing with one species, and in fact a lot of people thought we are dealing with a species complex, so we wanted to investigate that. We wanted to determine if P. rubi isolates vary in their ability to cause disease to raspberry, and we wanted to know if they differ in their susceptibility to fungicides. So what we did, or what Jane did, is she visited raspberry fields all up and down the West Coast, California to northern Washington. She collected plant material, and we always preferentially chose plants that were showing symptoms of P. rubi to increase our likelihood of finding it. When we got this material, we looked at it in 4 different ways. We did like Jerry did, we put cane pieces from the lesions on Petri dishes to see if the fungus would grow out. We did some root isolation, same thing, put them on Petri dishes. Soil baiting, this is where we use, similar to what Wendy was talking about, just kind of this container system where you put the inoculum in, put a raspberry plant in, and see if disease happens. And finally we used a DNA-based method to detect it in roots. So the pictures on the right kind of show some of the methods that we used, and the table that I'm showing you here, on the far side, you can see that we had 3 locations in Washington, 3 in Oregon, and 2 in California. And then across the top are the different isolation detection methods. And what I really want to point you to, first of all, is as far as scientists, what this showed us when we looked at these kind of, the first 3, the root, cane, and bait methods for detecting P. rubi. So these are kind of hands-on traditional, what we would call traditional methods. Our cane method did a lot better job of detecting P. rubi in these samples. And that's useful for us because I think it will just, increase our ability to be able to detect it and understand this pathogen more. The really interesting thing here, and it kind of goes back to the same stuff with Jerry and his verticillium assay, is that when we used a PCR method or a DNA-based method, you can see the 3 traditional, what I'm calling traditional methods, the maximum detection that we had was about 40%. When we used a DNA-based technique on these same samples, all of a sudden the incidence of this disease increased to 70%. So we were missing a lot using Petri dish traditional type methods. So we now, in the future, are going to move towards pretty much PCR-based tests, I think, for detection of this disease. So the other thing, so our second objective is we want to know what Phytophthora are present in raspberry. And this came out of some of the work that Craig McConnell did years ago, where he did his Phytophthora survey. And he went in and they got samples pre-plant in field, kind of just shows you the samples that he collected. And not only did he come up with positives for Phytophthora rubi, but he came up with a lot of positives for Phytophthora species. And those were really never identified the species. So kind of at the end of all of Craig's work, it was kind of like, well, I think we might have a complex of Phytophthora that are causing raspberry root rot. And in fact, other Phytophthora species have been reported on raspberry, and they're listed up there. So we were interested in looking at this further. And so what Jane did is when we went to all those fields, so there were 346 isolates collected from western state raspberries, she went in and she used DNA-based methods to identify them, and 99% of them were Phytophthora rubi. In fact, only 1 isolate out of the 346 was something else, and that's that Phytophthora bischeri, which actually is a first report on raspberry. But I think this, at least from our experience and what we show here, Phytophthora rubi is the Phytophthora that is causing root rot in raspberry in the western United States. Jane went further to look at some of the diversity which exists among these Phytophthora rubi isolates. She did some more DNA-based work on it, and essentially what she found across the western states is that genetically, there's no difference in these organisms. So it's pretty much, so the conclusion we came to was, everything in the western states as far as Phytophthora causing root rot is similar genetically. Third part of our study, we want to know if P. rubi isolates differ in their ability to cause root rot. And the way we did this was in our container system, similar to what Tom Walters has used to assess some of our treatments in our field trials. Containers, we'll just go in, we put a soilless mix in, we add inoculum, put a meeker plant on top. 4 weeks later, either you get disease or you don't. When we took it down, we did root rot ratings. That's shown on the right. Up in the top corner are healthy roots, long, nice white roots. Down on the bottom, 12, that's a root system that's severely impacted by Phytophthora rubi. We looked at root length and top biomass. And so our results, similar to the genetic diversity that exists within Phytophthora rubi, there's no difference in pathogenicity of the isolates we looked at in their ability to cause disease to raspberry. And that's shown here in these two graphs, which I believe the top one is root length, the bottom's foliar weight. The bars furthest to the left are our controls. We always include two controls in these studies. We have a flooded and a non-flooded one. Again, going back to the life cycle, we know that Phytophthora rubi likes really moist conditions, so we create an environment that's conducive for this organism to cause disease. And if you look, so the two controls had greater root length, more foliar dry weight than any of the plants that were grown and exposed to the isolates. And we had isolates from California, Oregon, and Washington included in this assay. And just a few more pictures. This is kind of what we saw at the end. On the far left is a control plant. The other two were exposed to P. rubi, and then a kind of a side-by-side comparison of a plant that didn't have P. rubi and one that did. So, pretty significant impact that this pathogen has on raspberry. And then finally, our last objective was to see if the isolates that we had collected from the western states differed in sensitivity to ridamil. It's a common fungicide that's used in raspberry production systems to control Phytophthora. And what we did here was we exposed our different isolates in Petri dishes, as shown here on the right, to different concentrations, and therefore we can figure out the concentration that's needed to kill 50% of the population. And we're getting a reoccurring theme here because I have a lot of no's on a lot of my slides, and essentially we found similar results to what we found in our pathogenicity trial and also our genetic diversity studies, in that there is no difference in sensitivity to ridamil among isolates collected from California, Oregon, and Washington. And that's shown here with you just look at the middle bar in those boxes, that's the mean, so there's really not much difference between the isolates that were combined for each of these locations. I was talking to some folks earlier about this, and I just wanted to put in as a side note, when we look at the isolates that we included from Washington, there's actually only 2 or 3 that are included there. And of those 2 or 3, I believe only one of them is from northern Washington. So I guess I say this because I don't think we've exhaustively looked at differences in sensitivity of isolates from northern Washington to ridamil, but certainly if you kind of take a big picture look at it, we don't seem to see resistance to ridamil in these isolates. So our conclusions from this current research, I think we can say with quite a bit of confidence that P. rubi is the most common Phytophthora found in western state raspberries. As I showed in the first part of our study, the molecular tools that we use are the most reliable means of detecting Phytophthora rubi, quite a bit more effective. If you remember, 40% versus 70% incidence, that's quite a difference. And then finally, that the Phytophthora rubi isolates collected from western states, they don't differ in their ability to cause disease to raspberry, and nor do they differ in their resistance to the fungicide ridamil. And And so just in conclusion, some future directions we'd like to go with Phytophthora rubi research. So that kind of wrapped up everything that Jane did during her year in Nick Grunwald's lab. And as we move forward, we're going to continue to explore the genetic diversity of Phytophthora rubi isolates. So as part of this big grant that we had with Tom Walters, we got enough money to sequence the entire genome of this organism. So now we're going to have the ability to kind of go in and be able to compare Phytophthora rubi to other Phytophthora species to start to understand some of the mechanisms by which this pathogen causes disease. So that's going to be ongoing. This coming winter, we're going to implement a quantitative molecular technique for the detection of Phytophthora rubi. As I said before, we have compelling evidence that indicates this is the direction we need to be moving in. And so we're just going to take this a little bit further in a graduate student project. And then finally, we're going to determine the spatial dynamics of P. rubi in northern Washington fields with root rot. So the idea here is to be able to go to a field and start to understand some of the field components of the variability within a field that drives disease. I mean, you guys know that if you're in an area that gets flooded commonly, you're going to have root rot. But are those the only variables that are conducive for disease, or are there other variables within a field? And can we identify those for you to help you guide management practices? So with that, that's all I have on the root rot stuff. Are there any questions? Tom?
I guess the big question from a grower perspective on that is the possible ridimil resistance and the implications here. It'd be really good to be able to sample from some fields that have had a lot of ridimil use compared to some that didn't, to be able to zero in on that question. Would that be possible?
I think so. I think in combination with what Jerry's doing, Jerry, as you saw in his previous slides, in a lot of his survey stuff last year, he came up with additional P. rubi isolates. Maybe we can get the histories on those fields and kind of get an idea of which ones have a history of ridimil, which ones don't, and then do these same type of Petri dish assays. So I think that's very achievable in the short term.
Yeah, I think that could really influence dynamics in the fields if we find out whether that's really present or not.
Yeah.
Can you hear me now?
I can.
The nematicide fraction of the fumigant mix was L3D.
1,3-D.
1,3-D.
1,3-dichloropropene.
Thanks.
I always say it's good to be a nematologist because we have like 5 compounds I have to remember, right? So, unlike those weed scientists and entomologists who have a gazillion. So, are there any other questions? All right.